Good afternoon, class. Good afternoon. So today we're going to be talking about water and the process of osmosis, building on what we learned about last week with diffusion. So by show of hands, who here likes water? Anybody? Few people? Few people? That's good. I used to hate water. I used to only drink milk and soda. But as I grew up, I realized that water actually is pretty important. Now all I drink is water and iced tea. Lemon water sometimes too. So for today's lesson, we're going to be going over the importance of water, the characteristics of water. We're going to be talking about the process of osmosis and why osmosis is important in biology. So, who here has heard of osmosis? Few people? Few people? Okay. To start things off, we're going to do an activity called an idea toss. I want you guys to write down on a piece of paper what you think osmosis is, or a few keywords about osmosis, if you know a few keywords, or a sentence or two at the most. Write down what you think osmosis is, or any preconceptions you have about it. Once you're done, I want you to crumple that paper up and try to make it in this basket right here from your desks, okay? I'll give you guys like a minute and a half to write down something and then get ready to toss your papers in, okay? Okay guys, now that you had time to think about it, I want you to toss your papers to the basket and see if you can make it in, and we'll go over these, alright? Oh, nice try, nice try. Keep going, let's go. Oh, that one almost made it. Look, come on guys, you can do it. Gotta concentrate. Oh, we have a winner, good job. Let's see, any more? Come on guys. We're supposed to make a few. Alright, is that all of them? Alright, let's see these. Might have to go for some of the ones that weren't winners. Let's see here. Alright, so one of our winners, we have water, evaporation, hydration. Alright, that's some good ideas. So water is definitely related to osmosis. Osmosis deals with water. Evaporation and hydration, kind of, sort of. Osmosis is definitely involved in keeping your cells hydrated. They always tell you, keep drinking water after you've been running and sweating a lot, because some of your water is lost to evaporation. Here we go. Water, liquid, and slow. Yep, well, it definitely deals with water, like we said. Water is a liquid, and it is a slow process. Let's see, what else do we have here? And another miss was water, and it's related to diffusion. Yes, this is correct. So, a few key ideas we have is we know it relates to water, and we know it relates to diffusion. Anybody else have any ideas they want to add to that before we actually get to the actual definition of osmosis? Let's see if any of these guys have. No. Water. I have no idea. Okay, well, hopefully after this lecture you'll have an idea. Alright, so let's get to it. Well guys, I have a little picture example for you of what osmosis actually is. It's here's the definition. The movement of cats through a membrane into a region of lower cat concentration. Alright, yeah, but just a helpful visual representation in a funny way. As you can see, like, if you think of the beds as two cells next to each other, the cat is something in the water. But what they have crossed out here is the movement of solvent molecules through a membrane into a region of lower or higher solute concentration. Higher solute. We'll get into that right now in the following slides. Alright, so let's talk about the importance of water for a little bit. If you guys didn't know, water is very important. Most biological processes and most chemical processes only occur in water. Just rubbing dry ingredients together won't get you too far. So it's essential in the transport of materials inside an organism at the cellular level. Water is very important. And I mean, think about it. Human beings have water coursing through us at all times. Any ideas what we call that? Blood, right? Exactly. Blood is composed mainly of water. In order to survive, you need blood to circulate throughout the body, and a significant amount of blood is actually made out of water. Humans are roughly 60% water overall. Just thinking of the brain and the heart are 73% water. Okay? And so, we all live on this planet here called Earth. It's funny that they called it Earth because they really should have called it water. This planet is made of about 71% water. And land only makes up the other 29%. So, moving on. So along with water being important to animals, water is also very important to plants. Plants absorb water through their roots, and along with the water comes with a bunch of minerals and nutrients that they're reliant upon in order to grow healthy and strong. So looking at some of the basic characteristics of water, water is a polar molecule, or a polar compound, if you will. It's made up of 2 hydrogen atoms, 1 oxygen atom. Okay, and they're covalently bonded to each other. The shared electrons between the 3 molecules that make up water, or the 3 atoms that make up water, they aren't shared equally. Because oxygen is a bigger atom and more positive than hydrogen, the 2 hydrogen atoms attached to it, the electrons want to hang around the oxygen atom more than they hang around the hydrogen atoms. So, by show of hands, or who knows first, what kind of charge is associated with electrons? Think you know? Negative. Negative it is, right? So if the electrons are hanging around the oxygen atom more than they hang around the hydrogen atoms, what do you think is going to happen to the charge on that oxygen atom? Going to be negative, slightly negative, right? Exactly. Therefore, the charge on the hydrogen atoms are going to be slightly more positive because the electrons are hanging around there so much. Because of this pull and uneven distribution of charges, it results in a polar molecule. This is what we mean when we call a molecule polar. It has a positive end and a negative end. So key rule of thumb to remember is polar likes polar. Salt and sugar are both polar molecules. So, when you put them in water, they tend to dissolve very easily. That's because polar likes polar. So what happens when you put oil in water? Does it dissolve and does it mix very easily? No, right? That's because oil is a non-polar molecule, or a non-polar substance. So water will stick, but it is not sticky. What do we mean by this? Well, because water molecules have a positive end and a negative end, there's going to be an attraction between water molecules. This results in hydrogen bonding between the oxygen atom and the oxygen atom of one molecule and the hydrogen atoms of another molecule. This attraction leads to capillary action, and this capillary action is what we can see in the lab when you see liquids in water creep up thin tubes. And so water sticks to itself, and so if you use like a straw, you can suck water up of a straw, or if you use a really thin capillary tube, just by dabbing some water or some blood. Normally when you use capillary tubes to do testing, you will see it actually just shoot right up that little thin tube because it actually sticks to itself. But again, it will stick, but it's not sticky. When you put water on a wall, and then you try and put a ball or something to the wall and try to stick it to the wall via that water, it doesn't stay there. So that's what we mean when we say it's thick, but it's not sticky. Water is very resistant to temperature change. So when you try to put water on the stove and boil an egg, or if you're going to cook some rice or something, it takes a while and a lot of energy to heat that water to a boiling point. Water likes to resist temperature change. And finally, water expands when it freezes. So for those of you who didn't know, when you freeze water, it's definitely never a good idea to fill a glass cup up and put it in the freezer. Why? Because water will expand when it freezes, and you end up breaking that glass cup. Okay? So, just expanding on osmosis in everyday life. Have you ever gone on a trip? Any of you have a garden and forgot to water your plants for a few days, and you come back and you find, a wilted mess? This is because of osmosis and the point of water again. Like we said, plants absorb water from their roots, and so when you forget to water them for a few days, this water ends up going the opposite way. It leaves the plant cells, causing them to lose the internal pressure called turgor pressure that normally supports the plant upright, and they start to wilt. So the way you look at this has to do with evaporation, it has to do with osmosis, it has to do with the solute concentration inside and outside. So as the water outside the cells drops due to evaporation, the quantity of ions and other particles remain the same, so the ratio increases in solute concentration, and water wants to leave those cells into the intracellular space to make up for the lost water due to evaporation. Key part of osmosis that we haven't talked about yet is the semi-permeable membrane. So what am I talking about when I talk about semi-permeable membrane? Semi-permeable membrane is a membrane that allows some molecules to pass through them while blocking the passage of others. So those type of membranes are considered semi-permeable. So with osmosis, we're talking about membranes that have pores large enough for water molecules to pass through them. They need small enough to block molecules in the solid. So water is going to flow from the area with a smaller concentration of solutes to the side with a larger concentration of solutes. I know from the people that are going, wait a minute, what happened? It's going to be from high concentration to low concentration. Well, it is. It's going from a high water concentration to a low water concentration. Water is still flowing down its concentration gradient through this semi-permeable membrane. You can think of any examples of semi-permeable membranes that you have around your house that you've seen before? Any examples? Yep, what do you got? Okay, yep, that's a good example. Not the outside, right? Yeah, the inside. So we would remove the outside hard shell of an egg. Not just like dissolve that hard shell of that egg, you left with just an inner membrane. That is a semi-permeable membrane. So they have made semi-permeable membrane with really well known dialysis tubing. That's why we have to let water go through while water passes through the molecules. And so in a simple solution containing two substances that are evenly mixed together, the solid is the substance to be dissolved, and the solvent is the one doing the dissolving. So in most cases, water is going to be the solvent that is doing the dissolving. So for example, if you have a glass of salt water, what is being dissolved? Salt. What's doing the dissolving? Water. So salt through the solid, salt through the water, and you have now made your compounds of your surface solution. So, the actual definition of osmosis. We have the spontaneous net movement of solvent molecules like water across a semi-permeable membrane from a less concentrated solution into a more concentrated solution until an equilibrium is established on each side of the membrane. Now remember, from last time, equilibrium, we're talking about dynamic equilibrium. Unpacking the net movement, well, they're saying that once this equilibrium is achieved, solvent molecules are still going to be moving back and forth, but there won't be any net change. You won't be able to see any real difference between them once this equilibrium has been established. And again, semi-permeable, we just went over that. And less concentrated, remember, we're talking about more water is considered less concentrated in the solvents. So when you have more water, low solutes, high solutes, you have less water. Water always wants to go to the area where there's less water. It wants to move down its concentration gradient. So a nice rule of thumb to remember, osmosis is literally the diffusion of water. Whereas diffusion, you're talking about solutes. Osmosis, we're talking about solvents. And so spontaneous also is in here. We're talking about the spontaneous net movement. This is to remind you guys that osmosis does not require energy. Just like diffusion, osmosis is a passive process. It just occurs. So this is, again, remember, due to the second law of thermodynamics, where everything's in constant motion. So osmosis and diffusion are both passive processes. So, got a question for you guys. Here we got a picture of a plant cell. And it normally lives in fresh water. If I were to take this cell and place it in a beaker of 25% salt water solution, that's a lot of salt water. Okay, I know plants, have some salts inside of them, but nowhere near 25% salts. And especially this plant that lives in fresh water versus like a plant that lives in the ocean that lives in salt water. So this is a freshwater plant. And I put this cell in a beaker of 25% salt water. What do you think is going to happen? A, B, or C? A is going to increase in size. B, is it going to decrease in size? Or C, is it going to remain the same size? So take a minute or two to think about it. Write down your answer. And then I want you to pair up and discuss your answer and see if you and your partner have the same answer or if you have different answers. If you guys have different answers, try to see if one can get the other to switch answers, or if your reasoning behind it is just too good and you guys both want to stick with your original answer. And then we'll talk about it after you guys are done talking with each other, okay? Okay, you guys figured it out? Convinced everybody of the right answer? So the actual answer is going to be B. It will decrease in size. Now why is that? Well, as you can see, most of the plant cell is made up by this central vacuole, and it's a good majority of it. And the central vacuole holds the water inside the cell. So remember, water likes to go where there are more solutes. In this case, we're talking about salt being the solute. So where is the more salt? Inside the cell or in the 25% solution outside the cell? Outside the cell, right? So water is going to want to go outside the cell to try and balance out the two osmotic pressures on each side of that cellular membrane, thus decreasing the size of the cell. So, same cell, same plant cell. Suppose you killed that plant cell in the picture to the right and then placed the dead plant cell in 25% salt water solution. First we had a live plant cell, now we have a dead plant cell. Remember, this plant cell used to live in fresh water. If it was alive, it would prefer freshwater environment to salt water. So, what do you think is going to happen with this dead cell? A, osmosis would not occur. B, osmosis and diffusion would continue to occur. C, only diffusion would occur. Or D, only osmosis would occur. Again, I want you to pair up and talk about your answers, and then we'll discuss it together as a class, all right? Okay, if you guys decided on answers, show of hands. Who here thinks A? B? C? And D. I know D isn't up there, but D if it was. Okay, the answer is B. Osmosis and diffusion diffusion both continue to occur in a dead plant cell. This is a major misconception. Most people think that once you die, osmosis and diffusion stop. Because why would they continue going? Your body doesn't need it to happen, plant cells don't need it to happen, they're dead. Well, remember from the last slide, we said that osmosis and diffusion are both spontaneous processes. And remember, spontaneous means they require no energy to occur. So since osmosis and diffusion are both passive processes, at least for a little while after you killed this cell and put it in 25% saltwater solution, water is still going to pour out of there until either it's done with all the water available or it reaches some sort of equilibrium to where the concentration gradients are very similar and water stops going in. So the rate of osmosis varies. Again, just like diffusion rate varies, osmosis rate can also vary. And mainly osmosis is dependent, or excuse me, the rate of osmosis is mainly dependent on the same things that the rate of diffusion was dependent on. Except with osmosis, there are a few extra. So we're still looking at the difference in the concentration gradient. We're still looking at the distance between two regions that these molecules have to travel. Temperature still plays a part, and the size of the molecules in question still play a part. But in addition, we're also looking at the size and the composition of the semi-permeable membrane that is in question or in the system. Like we're talking either the egg membrane or the dialysis tubing. Depending on the membrane in question, there's going to be a different number of pores, and the pore sizes are going to be different. And so that's unique to whatever substance you're talking about. And so these are the things that affect the rate of osmosis. You have bigger membrane size and bigger pore size, osmosis will happen a little bit faster. If you have smaller pore size, it'll slow it down. So just to elaborate a little bit more on this and drive it a little home, remember we're talking about water moving down water's concentration gradient against the concentration gradient of solids. Remember, it wants to go to the higher solid side. So in this picture, we have a concentrated sugar solution on one side and a dilute sugar solution on the other. What's that mean? We have a lot more sugar on the left compared to the sugar on the right, and the blue little dots you can imagine are water molecules. The red dots being the bigger sugar molecules. And so this partially permeable membrane, this semi-permeable membrane right down the middle, it allows the passage of water molecules because it has small pores, but it actually blocks the passage completely of sugar molecules. The sugar molecules are just too big to pass through. So given enough time, even against the pull of gravity, water will force this solution level up, while this solution level drops as water leaves it through this partially permeable membrane until this dynamic equilibrium is established. It wants to try and have as many, the ratio of water molecules to sugar molecules on this side equal the ratio of water molecules to sugar molecules on this side. And as you can see, there's only 3 sugar molecules on this side. So it has to really work to try and get those sides equal compared to the, over 10 or 12 so molecules on the other side. So we're moving from an area of low solid concentration to an area of high solid concentration. It's still moving down water's concentration gradient. So cell membranes also affect the rate of osmosis, and that's because they're very intricate. You got a whole bunch of intracellular membranes, aquaporins, proteins, and they all have different sizes and such. So here you have an example of your integral membrane proteins versus just your protein channels. You have glycoproteins attached to some of the carbohydrates. So the composition of cell membranes and the way lipids and proteins are structured is very important in determining the rate of osmosis. The number of aquaporins, the more aquaporins you have, the faster water can get through these cell membranes. But remember, water is polar. Cell membranes are phospholipid bilayers, so they also have a positive and a negative head or negative end. So that's why water is able to get through these membranes. So temperature, again, also has an effect on the rate of osmosis. Whether you want to speed it up or slow it down, you know how to control the temperature accordingly. So higher temperatures increase energy. We should all be familiar with higher temperatures and heat increasing energy, lower temperatures, and the loss of heat decreasing energy from a system. So when you increase the energy, you're therefore increasing the movement of molecules, and therefore with the increase of the movement, you're actually increasing the rate of osmosis and those molecules trying to establish that equilibrium through the membrane. If you're to lower the temperature, you're going to decrease the energy, slow down those molecules' movement, and decrease the rate of osmosis. So, cold water will take a lot longer to reach this equilibrium if you had a difference in osmotic pressures than hot water. So here you guys go. We're going to do an activity called speed dating. I want you to pair up, talk about your ideas, and then after about 2 minutes, I want you to switch, find a new partner, and talk about your ideas for another 2 minutes. And here's your question, what you guys are talking about. Why do patients at a hospital who are sick or are there for treatment or surgery and such, why do they receive IVs with a saline solution? For those of you who don't know what a saline solution is, it's basically a salt water solution. So again, we're going to do speed dating, pair up, talk about it, and then switch after 2 minutes, okay? And then once you guys are done talking with 2 different dates, we'll talk about it as a class. So after your guys' dates, what'd you come up with? Any guesses? Any guesses? What do you got? What do you got? All right, let's go over this. IVs are given to patients to keep them hydrated. You don't want patients getting dehydrated in the hospital. Well, how do you do that? You do that with this saline solution. We can't just give patients pure water because then you're going to be putting these cells into a hypotonic solution. You're going to have water having almost no salts in it. It's going to be pure water, like you said. So water's going to want to move from the area of low salt concentration to the area of high salt concentration. Well, even though we don't have that much salt in our red blood cells, there's some. So if given enough time, water will force its way into our red blood cells and cause them to burst. And that does not bode well for patients in a hospital because they definitely need their red blood cells to be healthy, transporting oxygen throughout their body. So when you think, okay, well, shouldn't a salt water solution cause the opposite effect? Shouldn't it cause, the water to leave the cells in like a hypertonic solution? Well, you'd be correct. If you have too much of a saline solution, too concentrated, water will end up leaving the blood cells and leaving the little bit of salt that's actually in the cells to try and balance out the major salt of that salt water solution. And so when this happens, we call this red blood cell crenation. They no longer have that biconcave shape. They no longer can bind to oxygen in the way they're supposed to. And that also is pretty bad news bears for our patients in the hospital. So what we want to try and establish is this isotonic solution right here in the middle, where our red blood cells are the happiest, where water is not only coming out, but also going in at the exact same rate. And to do this, you need to have the right concentration of saline solution. So again, you can't have pure water, but again, you don't want it to be too concentrated of salt water. You want to have just the right saline solution, anywhere between 5 to 10%, so that you'll create this nice isotonic solution that red blood cells will thrive in. Okay, and so once we have this isotonic solution and we have established this dynamic equilibrium, osmosis is still taking place, but it doesn't take place at any noticeable, with any noticeable net change. Okay, so the molecules are still moving. Solvent is still going from one side of the semipermeable membrane to the other side, but it's doing so at an equal rate, at an equilibrium. So once this equilibrium has been reached, the concentration gradient no longer exists in the system or the environment, okay? And remember, the molecules are moving randomly in all directions. There's just no noticeable net movement. And at any given moment, at any time, you can still have the equal probability of two molecules colliding with one another on both sides of the membrane. And so just to give you guys a demonstration, a quick video about what happens with osmosis in plant cells. There's going to be these two carrots here in this short, quick video. And again, I apologize, there's no sound. But which carrot is in salt water? What do you guys think? So we have carrot A. It's looking pretty flimsy. It's limp and easily bends. And this is after soaking for about 24 hours. Versus carrot B. Firm and crisp. Doesn't want to bend at all. Again, carrot A, you can almost bend it in half. Carrot B, very firm, very crisp. So what do you guys think? So now that you saw the video and the characteristics, I want you guys to pair up and we're going to do some idea coaching. So one student's going to be the coach while the other student is going to be the student. And so the coach is going to coach up that student and explain to them what happened and how they justify it. And then after the student is done coaching, we're going to switch roles and have the student become the coach and the coach become the student. And you'll explain it back and see if your guys' explanations match up or not. And so once you guys are done talking about it, we'll talk about it as a class. So get to coaching. All right, team and coaches, what do you guys figure out? Is the carrot in A that's all flimsy and malleable in salt water? Or is the carrot in B that's all stiff and firm in salt water? What do you guys think? Good job, it's A. That's right. So the water over the 24 hours went from the inside of the carrot cells to the outside of the carrot cells, thus leaving those plant cells with a lot of space in them, and so they're able to move them around and bend pretty easily, versus the carrot in B that had the water rushing into it over the past 24 hours. Those central vacuoles are really, 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 really tight, and so you can't bend them at all. That's why it's really stiff and unmalleable. So what questions do you have about this lecture?